Okay, in that first part of meiosis, we looked at meiosis 1, big things in meiosis 1 like crossing over, how tetrads form, the whole purpose of that. And remember, in meiosis 1, we ended with two cells that have 46 chromosomes in each cell. So now we're going to look at meiosis 2. In meiosis 2, we're not replicating the DNA anymore. We're just starting out with those two cells that were made in meiosis 1, and remember each one has 46 chromosomes. So here we go. Now with meiosis 2, we're starting off, just like through meiosis 1, we're starting off with prophase 2. Prophase 2, we have those two cells that were made in meiosis 1. The nucleus dissolves again, disappears. So the nucleus is disappearing. Now we have a chromosome from mom, we have a chromosome from dad. Remember that they have been replicated back in right before meiosis 1 started, so they have sister chromatids. Spindle fibers form. Remember, each cell has 46 chromosomes in it. No replication occurred. Now in metaphase 2, just like last time, your chromosomes line up along the middle. Then in anaphase 2, the sister chromatids are pulled apart at the centromere and they are pulled to opposite ends of the cell by the spindle fibers. And then in telophase 2, you have the division of the cytoplasm. You have the formation of a nucleus around the DNA that was pulled to each side of the cell. And an end result is going to be not 2, but 1, 2, 3, 4. After cytokinesis, 4 cells at the end of meiosis 2, and they are all haploid, which means they have half of the original amount of DNA. Okay, so each cell started out at 46 at the beginning of meiosis 2, and now at the end of meiosis 2, you have 4 cells, and they each contain half, or they're haploid. So here's a nice picture, just like the picture we looked at in meiosis 1 for meiosis 2. So here you have the two cells that we ended with at meiosis 1. Here they had 46 chromosomes in each cell, and then they go through the process of division again. Okay? And then the end result, at the very end of all of meiosis, when we're done with meiosis 1 and meiosis 2, is going to be 4 cells. They are genetically different because of the crossing over, because of independent assortment. We're going to look at that in a little more detail in a minute. And they are haploid, which means they only contain 23 chromosomes. So 23 chromosomes here, 23 here, 23 here, 23 here. Now we know that they are different, and there are 3 main reasons why. Okay, and we're going to look at these 3 mechanisms that contribute to this genetic variation, this genetic diversity. It is very important because if not, then every child that was produced from parents would be exactly the same genetically, and then that would lower genetic diversity or genetic variation, and we'll get into why that's going to be a problem. We'll talk about that a little bit later. But we're going to look at the 3 genetic reasons or genetic diversity or genetic variation reasons or mechanisms that this is going to occur. The first one is called independent assortment. Now I mentioned when we first started looking at meiosis 1 that in metaphase 1, independent assortment is going to occur, which means that when the chromosomes line up along the middle of the cell, they're going to line up randomly, which means that some of dad's chromosomes are going to line up on one side, some of mom's are going to line up on the other, but it's going to be different. It doesn't mean that all of dad's chromosomes are going to go to one cell and all of mom's chromosomes are going to go to another cell when that cell divides. It's kind of like a flip of the coin. Some of dad's will go one direction, some of mom's will go the other. So this mechanism or this genetic process of increasing diversity in our genes, this independent assortment is very, very very key, a key integral part in making sure that that occurs. It happens in meiosis 1, it happens in meiosis 2. Once again, the big thing is if they're going to be a billion people, that each one of those sperm and egg that are going to produce those billion people are genetically different. And next one, crossing over. We talked about crossing over. We know that happens in prophase 1. We know that the process through synapsis where the two homologous chromosomes pair up, okay, to form a tetrad, that's going to allow them, once they form that tetrad and crossing over to occur, that's going to allow them to swap pieces of their genetic material. Some of mom's and dad's genetic pieces get swapped to another chromosome. Once again, that just increases genetic diversity. So now when the cell splits and that DNA splits, some of it goes to one side, some of it goes to the other. That doesn't mean that all of dad's chromosome is going to go to one side and all of mom's is going to the other, because there will be pieces of each that are going to be swapped. So it's just increasing genetic variation. I keep saying increasing genetic variation. Why it's important, and the main reason it's important is because the lower the genetic diversity, the more problems with mutations, genetic disorders, life expectancy, all of those things occur, lower life expectancy. So you have increase in gene mutation, you have increase in genetic disorders, you have lower life expectancy. Okay, and the reason is because if you're swapping the same genes over and over and over, our genetics can't be built that way. Our DNA is not built that way, and then codes and copying of those codes tend to have higher problems with that, and so this is where our genetic mutations and disorders come from, or it can increase that. Okay, so let's look at this last one, this random fertilization. Talked about joining of two gametes produced independently. Okay, now the process of producing sperm is called spermatogenesis. It's where sperm are made. Okay, you start out with a diploid cell. It goes through meiosis twice, meiosis 1, meiosis 2. Then you have 4 sperm produced. And oogenesis, the production of female gametes, or the eggs. All right, so what's happening here, and this is just, remember, males produce thousands of sperm in a lifetime. Okay, females only have a certain number of eggs that are produced within a lifetime, and to make sure that differentiation occurs to the highest level, there's only going to be one, even though there are going to be 4 cells that are produced at the end result of oogenesis, only one is going to be viable, which means only one is going to get most of the cytoplasm and become the one that is going to be used as an egg or an ovum, and then the other ones kind of are byproducts. Okay, they're called polar bodies or byproducts. They get dumped and recycled. They're not used. So you only get one viable ovum that works at the end of oogenesis, or the production of your female eggs.